All right, this is Kelly Eversole, and I'm the Executive Director of the International Alliance for Phytobiomes Research, and I'd like to welcome everyone to our webinar series. I'm going to give you a brief overview of the Phytobiomes Alliance and then lead into our webinar. So we are a nonprofit consortium comprised of industry, academic, and governmental scientists who are located in the US, but we are international in scope, in membership, sponsorship, and in our research activities. Before I continue, I want to thank all of our sponsors who actually make it possible for us to have a webinar series and for us to coordinate our activities. So before we move into the webinar, I thought it might be helpful just to go through kind of an overview of what a phytobiome is, because I know there's often a lot of confusion about that. And phytobiomes is really thinking about the complex systems of plant-based agriculture. It's the idea of a plant in a particular biome or a specific environment. In fields, that's the soils, and in the weather, comprised of climate as well as water. It also includes the microbiomes, the macroorganisms and macrofauna, as well as arthropods, other animals, and plants. And all of these aspects are heavily influenced by management practices. And all of that and all of these interactions comprise the phytobiome. Our vision is to take all of this information and by 2050 give farmers the ability to use predictive and prescriptive analytics based on geophysical and biological conditions for their particular site to determine what's the best combination of crops, management practices, and inputs for any particular year. Our next Phytobiomes webinar will be on the 18th of June, and we'll be discussing and learning about a project that's been funded in the EU, and it is much broader than even plant-based agriculture, and it goes into animal microbiomes, food microbiomes, etc. So I think you'll find it of interest as well. And you can register at this link or on the Phytobiomes Alliance website. So just to let everyone know, the webinar is being recorded and it will be posted on the Phytobiomes Alliance YouTube channel within a couple of days. It just takes a few days to get the video ready. The presentation section will all be followed by Q&As, but the Q&As, please submit your questions into the Q&A panel. Do not use the chat button. You can use the chat panel to talk with fellow attendees or individually with any of the organizers. And again, but put your questions in the Q&A panel. And all of the handouts and the presentations, you can download those and by clicking on the handouts link. So without further ado, I'd like to introduce our leaders for the webinar today. Karyosoft Augmented Innovations is going to talk about unleashing your innovation potential through data management and analytics platform and microbial genomics. And we have the pleasure of having 3 people with us today, but I'm going to turn it over to Nassir Patel, the Chief Marketing Officer for Karyosoft, and he's going to launch the webinar. But again, thank you for your participation, and I'll turn it over to Nassir.
Thank you, Kelly, for that warm introduction, and hopefully you can all see my screen at the moment. Okay, So thanks once again, Kelly, for that warm introduction, and Phytobiomes Alliance for inviting us and providing us this opportunity for this webinar presentation. So as Kelly mentioned, what we wanted to cover in today's webinar is how data management and analytics can be used in microbial genomics to unleash your innovation potential. From Karyosoft, as Kelly mentioned, we've got Subinoy, who's our lead genomics scientist. We've got Rajesh, and we've got myself, Nassir. So with that, we'll move right into the presentation portion of the webinar. And I wanted to start here, which is really a big picture. And all over the world, scientists like yourselves are using next generation sequencing technology to understand microbes and organisms at the molecular level. And they're doing this, as Kelly pointed out, to solve problems, right? Primarily related to hunger or food security. And this is happening, as Kelly pointed out, not just in agriculture, but also in other life sciences verticals or other fields, and fields that use genomics data or next generation sequencing as the underlying technology. So with decreasing costs of sequencing, the application and use of next generation sequencing has really increased exponentially. And genomics data has proliferated, and the management and use and consumption of that genomics data has become ever increasing with challenges. So let's bring it closer to home for those that are participating in the webinar. You know, in the agricultural sciences space, yourselves, you might be working in the spaces of, just as examples, strain engineering, agronomic traits, gene discovery, perhaps biologicals for seed treatment as examples, right? So our intent today is not to cover all possible cases, but we'll go through several case studies to highlight how data management can be used. And I know I'm, in some sense, I'm preaching to the choir, because as users of genomics data, you're probably already experiencing those challenges. So hopefully this is very close to home. So I wanted to encapsulate the challenge or the problem that we're facing in this space. Right, if you look at the pictures at the top portion of the slide, right, today the view is, or the analogy we're going to draw is, that genomics data is organized and structured like the pile of books on the left, right? Very disorganized. And as you can imagine, if you're trying to find a certain book, or perhaps a certain quote within one of the books, in fact, if it's organized like that, you're going to spend all of your time trying to find the right book you're looking for, and perhaps the right paragraph or quote, right? And all your time gets consumed in the searching portion. Versus if the books are organized like depicted on the right, in a very structured and orderly format, it's very quick and efficient to find the book you're looking for and actually consume the content. And so whether you're doing, in this analogy, reading for pleasure on a Sunday afternoon, you get time to actually read the book and enjoy it. Or if you're pursuing an intellectual objective or business objective, you can actually consume that data and generate insights by actually working with the data and mining it, as opposed to looking for it and managing it. So this highlights the challenge here in front of us. And again, I know you're all facing it, but just wanted to encapsulate here to set the stage for what we're going to cover for the rest of the webinar. So the first challenge we've experienced working with our clients and customers is data repositories. And what I mean by that is there are multiple data repositories within an organization. So whether it's a university, academia, or a company, a scientist may be generating sequence data and storing it in a certain location. Other scientists or groups may be doing the same thing, perhaps with a different objective or different organism, and storing it in different locations. So what we've seen is the multiple repositories creates a challenge. Secondly, data formats. The challenge with data formats may come in several flavors. First could be, as an example, that the sequencing technology may be different and so renders the data in a different format. Secondly, data quite often is currently stored in a raw data format. And so when it comes time to mine or actually analyze that data, it's not easy. There's a challenge there before you can actually use or consume that data. The third challenge I want to highlight is this notion of private and public data collections. So again, whether you're in a university or a company, you're probably generating genomics data and storing it, and perhaps that's proprietary that you want to protect. But at times when you're doing your innovation work, there may be public data that you want access to or that you want to compare to. And today, there's no good way to bring together private proprietary data with public data and compare across those collections. So that presents another challenge. The fourth challenge I want to highlight is around the data mining and visualization tools itself. So many organizations, again, whether it be universities or companies, may have in-house developed mining or visualization tools, but in general, our observation or experience with organizations is they are developed for a specific application. And additionally, you may tend to want to use, connecting to the point up above, public data or publicly available tools and browsers. And that's not easy because those tools are either specific and disjointed or not connected and so not integrated. And again, that presents a new set of challenges. The last challenge I want to highlight here is around bioinformaticians' time. So in the R&D space, in the innovation space, this could be a constrained resource. And that's what we've experienced with many organizations and clients, that the rate limiting step in terms of the rate of innovation is quite often dependent on availability of bioinformaticians. And so if we can work around that or work through that challenge as well as the other challenges, we feel we can move our effort in this space on innovation from the left side of the page towards the right side. So hopefully that makes sense in terms of the challenges, and we're going to speak to some of these challenges here. The bottom part of the page is a depiction more in our world in terms of the multiple repositories, various data formats, public and private data collections, and trying to get that in one place. That represents the challenge, and our end aim point is what it looks like towards the right bottom. So if that's clear, and I know that all of you feel that those pain points in your daily workflow, right? You struggle with data access and managing that data. What we wanted to do in today's webinar and the remainder of today's webinar is cover 3 use cases. The first one is around gene discovery. The second one is focused on strain engineering. The third one is targeted at microbial discovery. Now, the intent of these use cases or case studies is not the case study themselves, but what we're trying to get at is the workflow and the efficiency lost or gained through a data management and analytics platform. So, as I said a couple of slides ago, you may be working in some different spaces than these case studies, but I'm sure you felt the pain that we're talking about here. So what I want you to think about as we're going through these case studies is your situation, your workflow, your struggles, and what we can connect to through these case studies in terms of opportunities for improvement and what a data management and analytics platform might achieve or deliver. So Subinoy is going to take us through these 3 case studies, and once he's done that, we'll come back to Lowside, the platform that is being used in the 3 case studies. And we'll give you a quick overview of the features of Lowside, and then look at the benefits and value that Lowside can deliver, and we'll wind up with a summary towards the end of the webinar. So if that all makes sense and is good, we can turn it over to Subinoy, who will take us through the case studies.
Thank you, Nassir, for setting the stage. I also like to thank Kelly and Isabu for this webinar opportunity. As Nassir mentioned, here I would like to share with you how our platform can help the scientist through 3 case studies. In this first case study, one of our clients was interested to discover a patentable gene from their own private microbial collections. However, there are many challenges. Number one, data privacy that Nassir just talked about. So due to privacy, organizations can't use their private data for query to the public databases. This is a major roadblock, especially for industry clients. So number two is creating and mining the patent database. So you need someone who can create for you the special patent database so that you can use. Number three, data dispersion over many repositories, both within and outside of organizations. So data usually kept in multiple repositories and are not properly maintained both within and outside of the organizations. And number four is most important, is the availability of the bioinformation. Suppose even the scientists settle for command line based tools and the mining the data with the help of the bioinformation, but the availability of the bioinformation time is a major challenge because bioinformation help multiple other projects and they work for the entire R&D team. So getting in time, the bioinformation time is very important and it's often don't happen. So as a quick note, for this case study, we are using the data from NCBI and not from any of our clients. So as you all know, in a typical workflow, the scientists prepare the microbial samples from their biological library collections and submit them to the NGS facilities for sequencing purpose. Once the raw data are assembled and annotated by the bioinformatician and store them on an internal servers and various other devices generally in raw format. And then scientists will mine this data for microbes containing pesticidal genes with the help of the bioinformatician. And then they conduct downstream lab work before testing it in the field. So in this typical workflow, according to the scientists who work on them, and also as per the NSF-sponsored study that we conducted earlier, this data access and mining step is one of the time-consuming part for the scientist, as often bioinformatics are not available due to their busy scheduling, and also they support multiple other projects in the organizations. And I, being myself a bioinformatician, I know that this data access and mining part is one of the most important part in this total workflow. And it is also very painful process or step in the whole workflow. So if you want to hire a bioinformatician for each process, for each data processing step, or each project, then it is also very costly affair for the organizations and the institute. So here, Lowside fits in very well in the data access and mining stage of your workflow by being it on-premise or on your cloud server. Lowside serves as a central repository. Nassir already talked about this. By unifying all your data in one place and connect the data to the mining process, analysis, and visualizations. You can do it in just a few clicks and thus free up the bioinformatics time. And also any scientist can also do by themselves. So Lowside being a web-based platform, scientists can access the Lowside using any modern browsers on any devices. And after logging into their secure environment, they can see the home page and the dashboard, which showcase the data they have and how they are organized. So in this study purpose, we downloaded the whole genome sequence data of about 135,000 bacteria from NCBI and arranged them into 2 major genome collections, namely public and private. So if you see the donut, the red one is the public and the orange one is the private. So in private group, we created 2 organism-specific sub-collections for BT and Spinosub. And I must say, this page is very much customizable and flexible. So you can store the data by projects or by organisms, etc. So all the flexibility is with you. And depending on the kind of projects or what you are looking for, you can add these data sets. Now to proceed with this project goal, click on the genes in the menu indicated by the hand symbol on this navigation bar, and that will take you to the dedicated genes page. So after coming to the genes page, first question to ask, how many pesticidal genes are there in their private collections? So on Lowside, you have the flexibility to search in all or specific collections. In this case, the scientist selects the private collections and search for all genes under the pesticidal descriptions. Now this search resulted in 2 pesticidal genes out of 3 in private collections. You can also download this sequence in FASTA format from the right-hand corner drop-down menu, as you can see in this screen. Now, the scientist has discovered that there are 2 pesticidal genes in their private collection. The second question to ask, are these genes patentable? To find the answer, click on the blast menu indicated by hand symbol on the navigation bar, and that will lead to the dedicated blast page. Now paste this FASTA sequence of both CRY genes and select the NCBI patent nucleotide sequences for multi-blast analysis. And for this pre-formatted database, we have downloaded all the available 99,000 patent sequences from NCBI. But as you know, that some of our clients actually mentioned that they would purchase additional patent database from various vendors. So if you have that additional database, patent database, then we can do the pre-formatted database, which you can keep on the Lowside along with the NCBI data. And that will broaden your search horizon. And creating of this pre-formatted database is totally customizable and client specific. So we, according to your need, we can totally customize this pre-formatted database. So here scientists also perform single, multi, and custom blast job using the combinations of different databases on Lowside. You have to select the appropriate program and name the job for future references, and then click the blast. It will lead to the My Task page. This My Task page is unique, and it serves as a user-specific repository for all previous studies that the scientist conducted. So when you will come back later, and scientists can search for all the previous study that they have done, and thereby you don't need to run the blast job again if you already run it before, and thereby you can save a lot of time and energy for the organizations. Now to see the results for a specific blast job, you click on here, and for these purposes you have the second one, and then you click here, and that link will lead to the visualization page. The results indicate that CRY1BA gene has already been patented, and also give the patent number, percentage identity, and other relevant information. Now to discover the patentability of CRY5AA gene, click on the scroll down button and select the CRY5AA. These results indicate that there is no patent sequence that showed homology to CRY5AA. So as a quick note, this analysis is done using 99,000 patent sequence from NCBI, but typically the patent search will be much broader, as I described earlier. So you need some proprietary patent database from vendors. And if you have that additional patent sequence, then you can perform those patent search through the Lowside as well. Now to determine the novelty of CRY5AA, run a blast against all the public database on Lowside by clicking the appropriate boxes and then do the blast. And these results indicate that the gene is showing homology to the whole genome sequence data of bacteria, where this gene is originated from. But basically, this is a self-blast test, so you're supposed to expect that kind of homology. But with this study, you can see what are the objectives we achieved. So here, Lowside helped them discover that there are 2 pesticidal genes in their private collections. So out of these 2 genes, one of them is patentable, and it is a novel candidate gene, which I guess is a very valuable information for the scientists. And this discovery will help them to decide whether to allocate and prioritize lab resources for both genes or one gene for all the downstream lab analysis. And as you see, all these discoveries are made in less than 15 clicks. So in this case study, we showcased for one gene. Now imagine if a scientist is analyzing hundreds or thousands of genes for getting the novelty and patentability informations. Considering the challenges that we mentioned earlier, Lowside can save weeks and months of scientific data mining time. And so they can start conducting their downstream lab and field work quicker and thus accelerating innovations. In a case study 2, it's a strain engineering. So another client was interested to discover mutations in a gene of their interest in a few bacterial states. For this case study, we choose RIB-F gene and 4 E. coli strains. So according to them, this typical challenge are the lack of user-friendly visualization tools, or even they are available, they are like standalone. So you just can't link them in a typical workflow. Now, to discover the mutations, a scientist needs several files, such as BAM, GCF, GFF3, and more. And it will be a labor-intensive process to collect all these different kind of files and process them and get the information of the mutations. But even say the scientists settle for command line based querying and mining of the data with the help of a bioinformatician, their availability is another big challenge, because the bioinformatician are busy and they're supporting multiple other projects in the R&D team. So that are the challenges you have to overcome to do this kind of work. As a quick note, for this case study, we are using data from NCBI and not data from any of our clients. So as I mentioned earlier, in this typical workflow, you require to have the BAM, GFF3, GCF, which are generated during the assembly and annotation steps. So as a bioinformatician, I know that collecting different files and then processing them is often very troublesome and challenging steps to go further and take out the meaningful information. So here, Lowside helped them to process all these steps. Now, to proceed with the project goal, click on the genomes on the navigation bar after logging, and we downloaded the whole genome sequencing data of representative multiple organisms such as bacteria, archaea, plants, protozoa, virus, fungi from the NCBI, and arranged them into two major genome collections, namely public and private. So we name them as public and private for better understanding. Here is a flexibility how you want to arrange. So we arrange them like 3 versus 3 into private and public. This page is customizable, and as I mentioned, so you can store data according to your need and the projects or the organisms. Now to proceed further with the project goal, click on the genomes on the navigation bar, which is symbolized by the hand symbol. Now after coming to this page, you'll see the search bar. So you search for the reference sequence data of E. coli strain that was used and click search. Now, since JBrowse is already integrated on Lowside, so no need to install any other JBrowse standalone applications or any other visualization tools. You just click the browse to visualize the E. coli reference genome through the integrated JBrowse. And you can easily visualize the nucleotide sequence and amino acid sequences. To add additional track, click on the track button on the menu. Now add annotation file in GFF3, reference SNP variant file, and BAM files of interested E. coli strains by selecting those files from stored location. Now you can add these tracks one by one or all together. Excuse me. Now adding all these tracks can be done by the bioinformaticians themselves, or give the file locations to scientists to add themselves, because it is very straightforward by adding tracks. Now on this browser, you have reference sequence, annotation file, reference SNP variants file, and the BAM file. And you can change the order however way you want to see them. Now to search for ribf gene, which is our interest in this big pile of genes, simply type the ribf gene here in the box and click go. The whole page of the browser with all the files will be zoomed in to ribf gene. And to know more about this gene, click on the ribf gene. So here are all these relevant information about the ribf gene. You can download the sequence by clicking on this button, which is symbolized by the hand symbol. And there are two different SNPs in ribf gene. To learn more about this SNP, you can click on the SNP itself. And the user can easily visualize the variations at genomic region at ribf gene. So here, if you look deeply, you can see there each vertical strip of color represents a nucleotide difference from the reference. In this case study, the user can detect 8 genomic regions that are varying from the reference. So with respect to reference SNPs, strain 1 is different from rest of the strain. To correlate the mutations with amino acid change, zoom in the reference sequence region. So in this case, strain 1, which has UGA at this region, which translates into stop codon. And however, the other strain have GGA that codes for the glycine amino acid. These results can be shared with your colleagues by clicking on the share button and send it through either email or instant messaging app. So in this case study, we discovered that there are 8 mutations in the ribf gene, which compared against the multiple strain. Now, one of the mutations clearly differentiates strain 1 from the other strain. Also, scientists could quickly correlate the mutations at the nucleotide level to amino acid and thus protein level, and share the whole message, whole information with the colleagues. So in this example, we use FASTA, GFF3, and BAM and BCF. But if a scientist wants to analyze with the big bed and big weak fields, they can also do that. So in this case study, we showcase discovery of mutations in one gene. Imagine if you have like multiple genes and how quickly mutations can be discovered. So Lowside make this complex mutation discovery easy for scientists under, say, 5 minutes. Now, the third case study, the microbial discovery. So in this case study, a scientist is interested to improve their understanding of coronavirus of their interest using the whole genome sequence data from NCBI within a secured environment. We choose coronavirus as a subject because it's a hot topic and everyone wants to know how we can use this case study to understand better, but you can use any other model organisms. But as a quick note, for this case study, we are using data from NCBI and not from any of our clients. Here is the project plan. So once the scientists sequence a coronavirus of interest, for example, MD334553, the next question comes in their mind if this genomic sequence is a novel or already known. If it is unknown genomic sequences, that means they discovered a novel virus. On the other hand, if it is a known genomic sequence, then it is not a novel virus. But either way, they'll be interested to know the genomic differences and their evolutionary relationship with other SARS-CoV-2 viruses. So now you got the sequence and very excited to know more about these genomes. Now to answer the questions, what next, we need some suitable tools and database so that can answer our questions. So for that purpose, Lowside can help in that. Simply click on blast menu on the navigation bar after logging into the Lowside. And here, the blast results will help to discover if this virus is already known or novel. Now come to the BLAST page and paste the new SARS-CoV-2 sequence of interest and BLAST against the in-house pre-formatted BLAST database of 928 SARS-CoV-2 genomes, which we created on Lowside, taking the data from NCBI. So this is a in-house pre-formatted database. It is not present other places, but we take the data from NCBI and create it ourselves on the Lowside. Now, these BLAST results will help the scientists to discover if the coronavirus of interest is a novel or known one. So here is the results of homology search of one SARS-CoV-2 sequence and with in-house pre-formatted SARS-CoV-2 database through the BLAST. And since we got the BLAST hit, so it is not a novel genomic sequence. Now we can discard the no part and more focus on the yes part. And then next we study the genomic differences and similarities with other SARS-CoV-2 viruses. For that, we did the multiple alignment of all these 928 whole genomes. And then we search where there are differences and similarities. We are particularly interested to find out the SNPs, and we found at least in 10 genomic regions that showed the variations when whole genome sequencing data of 928 SARS-CoV-2 sequences were compared. So if you see the consensus positions in the screen, so that indicates that there is the mutations and SNPs. And by digging deeper with the MT334553, when compared with the reference sequences of SARS-CoV-2, they differ only in 5 genomic regions, which are shown here. So if you look into the first figure, you will see that the consensus region, which is indicated by a tall bar, is actually absent on that particular location 8826 on the first figure. And here the nucleotide base changes from C to T, and that happened for other positions as well. So now the next step and the last step is to build a phylogenetic tree from the whole genome sequence data to determine the evolutionary relationship of MT334553 with other coronaviruses. So on the left, this is the complete phylogenetic tree of 928 SARS-CoV-2 whole genome sequences that were collected from the NCBI. And since the whole tree is very big, we keep it to fit in the screen, we keep it as a small one, and then we zoomed out the regions where this MT334553 are located. So it is indicated by this pink region. So that's where this particular genome of interest is located. And so if you see deeply, you will see that the phylogenetic tree indicates MT334553 is closely related with the MT259266 from USA Washington and MT308694 from USA Michigan. So currently, the multiple sequence alignment and the phylogenetic tree features are in developmental phase and not yet integrated on the Lowside, but still we wanted to share these features in this preliminary form. So in conclusions of this case study 3, scientists are able to discover the similarity, variations, and evolutionary relationship of SARS-CoV-2 of interest using Lowside. Now they can conduct similar studies for other microbes or organisms of interest. And with this, I'll turn the presentation over to Nassir. Once again, thanks to all.
Thank you, Subinoy, for taking us through the 3 case studies. So what we were going to do next is kind of move towards the wrap up here. I see we're going to be running short of time. So what I wanted to highlight here is, from the case studies, you can see how data management and analytics in the genomic space can help improve some of the efficiency and the results that you're looking for in terms of innovation. And, you know, as we indicated earlier, we use Lowside as the platform to do that. So we believe that Lowside's functionality can really serve as a linchpin to accomplish that purpose of efficiency and really helping you unleash your innovation potential. So we'll turn our attention next to Lowside itself, just a few highlights. And so first of all, what is Lowside? Lowside is a genomics data management and analytics platform. It was developed by Karyosoft Inc. And it can be deployed, as Subinoy said, either on premises within the enterprise or in the cloud. And so just some features that I wanted to highlight here is, and we've covered these in the case studies, but high data security. What does that mean? You know, because the application can be deployed either on premises or in the cloud, and you as an organization or as a scientist don't have to go outside into the public domain to mine data or retrieve data or tools, that helps secure the data. Secondly, data management. You know, Subinoy pointed out in several of the use cases how the data collections were organized. And if you remember the library picture that I started off with when I highlighted the challenges, right, this data management really means that as we set up and install Lowside, we can organize the data collections as you would like and suits your organizational style and needs, right? And what I mean by that is, for example, private versus public data, right? Or if you've got project-specific data, if you're working on fungi or viruses, those can all be separated into separate collections, but can be still mined or analyzed seamlessly. So data management's another feature. I also wanted to highlight the interactive search and with the integrated analytics and visualization functionality. And then lastly, Lowside is a web-based platform. And so what does that mean? It means two things. One, you can access Lowside through any device and really through any browser. So a lot of flexibility in terms of access because it's a web-based platform. So what does this mean in terms of value? So for users, such as yourself, if you're a scientist, I wanted to highlight these three things. And again, you've seen them in the use cases and we've touched on them earlier in the webinar. But first of all, data integrity. So there's no data prep required after the initial installation. We have a central repository, and so it eliminates data loss. Now, I just want to spend a few seconds highlighting a couple of things that come out of that point. Number one is Lowside, because of this central repository idea and no data loss, can really be your single source of truth for genomics data. So once it's in there, that's your repository of truth, right? No second guessing. Is there another collection somewhere else? Am I missing things? This is your single source of truth for genomics data. Secondly, I just wanted to point out a time aspect here. Once your genomics data is in this central repository, it can be stored there for future use. And that can be incredibly valuable because as you're innovating in the future, you can always use genomics data that was produced earlier on to compare against, right? As Subinoy pointed out, some of those comparative analysis. And just to drive that point home, in academia, in organizations, they store biological samples. Why is that? So as they're innovating in the future, they can draw back on these samples to compare against or use for innovation purposes. So the analogy here is the same, that once this data is in the central repository as the single source of truth, it can be used with confidence and going forward in future innovations. I also wanted to very quickly highlight, which we've already covered before, the quick, simple data mining with integrated analysis and visualization, right? A few clicks, a few seconds, and you've got your results. And the smooth integral workflow so you don't have to be bouncing around between applications. Now, if you do work in this space, you feel this pain every day or as you're doing your work, and hopefully these points make sense in terms of the value it can bring to you as a user. Now, if we think about this and think about what does this mean from an organizational perspective, right? In an organization, if there's groups of scientists that are working on different projects or in different efforts, this is what we wanted to highlight. So across groups or organizations, team efficiency can go up, right? Not just because of the efficiencies in the workflows that we've talked about, but because of eliminating some of the constraints of people who are doing certain parts of the work in the workflow. So team efficiency can go up across the organization. That translates to reduced R&D costs. And finally, probably the most exciting thing on this page is product discovery. The rate of product discovery can be increased. And so we put some numbers down at the bottom of the slide for typical clients or organizations that we work in the agricultural science space. If you focus on the efficiency or the cost aspects, in general, that can translate in the order of millions of dollars per year of value or benefits. If you look at it from the product discovery angle, and what we wanted to highlight is, imagine through this confidence and improved efficiency, being able to find that solution. And think about the book analogy, being able to actually spend your time consuming and generating insights from the data rather than fighting it and finding it, right? Imagine if you're able to discover and bring a product to market 1 year earlier, right? In typical ag organizations, that could be value in the order of hundreds of millions of dollars for the organization. So incredibly powerful. And that's what we're aiming for. Right, so just to recap here, what did we cover today? First, exponential increase in genomics data is happening. It's here and it's a challenge for you, right? In-house data management and analytics is a necessity. We can't keep going outside in a disjointed manner and compromising security. Lowside features that I wanted to highlight in summary: high data security, structured data management that meets your organizational needs. Quick, simple, interactive search with integrated analytics and visualization. So what does all that deliver? It eliminates the need for you as an organization to install multiple standalone analytics tools, and even eliminates the risk of compromising your data security because you have to access public data tools. So shown by the case studies, and I know we only had 3 in the time we had, but Lowside can be deployed in various applications, including orthology studies, to help you seamlessly analyze genes, proteins, genomes across multiple organizations, as illustrated by Subinoy. So at the end, what does this deliver? Lowside saves scientists time and can eliminate bottlenecks on constrained resources, and thinking about scientists and the workflow, right? And so through that, Lowside improves workflow efficiency, can reduce R&D costs, and can enable increased rate of innovation. And just remember that point about bringing product to market 1 year earlier. So what's next? I want to leave time for Q&A, but what's next? We focused this discussion today on microbial genomics because of the participants in the webinar. But we already have in the pipeline for Karyosoft, we've already worked towards a prototype of a natural languaging processing functionality. So what does that mean? You could query your genomics data using voice or natural language, English language, to query your data. Secondly, we're broadening the omics functionality to include other modules such as proteomics or transcriptomics. The third highlight I wanted to point out was that we're expanding data analytics and data analytics capabilities to other organisms, so aside from the ones we have in-house already. So with that, what we want to do is work with you to unleash your innovation potential. We know you and your organizations are trying to solve the world's most challenging problems, and we want to help you and do it together. So I'll quickly wind up so we have time for some Q&A and discussions, but I just wanted to acknowledge all of you participants today. hopefully you've been submitting your questions in through the panel. If you haven't, please do so now. We want to have a robust discussion. And if we don't have time to get everything answered today, we'll take that offline. I also wanted to thank the Phytobiomes Alliance members, and specifically Isabel and Kelly, for inviting us and hosting today's webinar. And then lastly, I wanted to thank our clients, Karyosoft clients. Not that we use their information, but some of the learnings and outcomes is what we're sharing. And then also the Karyosoft team members, not only the presenters today or the participants today, but the technical folks behind the scenes who put together and helped us put the case studies together. So with that, we can kind of move towards discussion and Q&A, but I did want to point out that our contact information is on the slide, info@karyosoft.com. So if we don't have a chance to answer everything, or you have further questions about Lowside, we know we just gave screenshots. So if you want a demo or want to explore or learn more or collaborate more, that's how you can get a hold of us or contact us. So with that, I'll turn it back to Kelly to moderate the Q&A. Thank you very much.
Great, thank you very much. Subinoy and Nassir, we're also going to be joined by Rajesh at this point, I believe.
Yes,
Correct.
Okay, great. It's always good to have the CEO involved as well. So we do have a few questions that have come in. One is, are SNP analysis only possible with high quality genomes? And if that is the case, is there a quality threshold that you use, perhaps in terms of number of contigs or N50 or another metric?
So Rajesh, want to answer?
Yes, that would be great.
Oh, okay. We basically, I mean, we showcased our platform as a use case study, taking the data from NCBI, but it depends on the data quality, what you generate, because we are not taking ownership for that quality of the data you are generating. But our platform can help you to determine the SNPs for you based on the quality of the data you input into Lowside.
So just to follow that up a little bit, one of the challenges with some of the NCBI data is that we don't really have a clue as to, in all cases, as to what is that quality. Does that affect this quality does quality actually affect the ability for the process to work?
I totally agree on that, because NCBI, there are some limitations with the data quality, but especially with the in-house data, when they are generating within the organization, they have a stringent value for quality of the data they generate. So if that data is put in Lowside, it will automatically give you the information. And also, if the quality metrics value needed to be shown on Lowside, that also we can inbuild, and that way the users will know what the quality of the data is for that particular analysis.
So based on what you've done so far, and again, this is just pushing a little more into this question, is there a recommended quality level that you would have for utilization of the platform?
We haven't basically, I mean, this varies from clients to client. Some clients wanted certain quality value. They think of that based on what they do internally. There is no gold standard from our experience when we are interacting with multiple clients. So it depends on what the individual clients want. So that can be used for the SNP analysis.
Could you give us a couple of examples without identifying your clients? I mean, part of the reason that I'm pushing a little bit on this is that, as you know, the Phytobiomes Alliance is really looking at developing standards, and standards both for research analysis as well as for future regulatory purposes. And by regulatory, I mean getting through the commercial, bringing products for commercial development. So just even having a few examples might be interesting as to what some of your clients are actually asking.
Once again, it depends on the project also. In sometimes when they have a 3x coverage of their sequence data, some they go up to 10x coverage for the sequence data to determine the SNP, to shortlist that sequence, shortlist the SNPs that they wanted to look into that. So this is the typical range what we have so far have come across. And again, like I mentioned, it is what the clients want in terms of that quality, which can be incorporated into Lowside. So we are not recommending anything what exactly what they want. It's what they want can be utilized as they wish. That is the angle I put it that.
Kelly, if I understood your question correctly, right? What you're looking for is kind of like an industry standard or recommendation, right? So I think the answer also is just to add to what Rajesh said. We haven't developed that or proposed that. Now with that question, maybe what we can do is take it offline and see if we can move towards some kind of understanding or thought around that, and then we do that offline and come back to you in the Alliance. So
I think that would be really helpful for any input that we can get on what makes the different platforms work more efficiently and actually deliver more efficiently would be helpful, I think. So exactly, I would appreciate that very much. Mm-hmm. And another question might be, kind of tied into that. You talked about NCBI and bringing the NCBI data in, client data. Are there other platforms that you can easily accept data from? Or does it matter? Is it agnostic in that regard?
In any other platform means, would like to understand what you mean by that platform. Is it like a sequencing platform? Is that what you are asking?
Yes. Or maybe it's not even a sequencing platform. Maybe it's for transcriptomics, and maybe that's part of. You mentioned you're in the process of developing some additional modules.
Definitely, yeah. At this point, we are agnostic of the sequencing platform. The data can come from Illumina or Roche or any platform it can come, because all we need is the FASTA and GFO files as an input file to get into Lowside. So it can come from anywhere. So it doesn't matter at all for us.
So it really is agnostic in that regard. So another question is, does the mentioned assembly or annotation pipeline need to be used by the client in order to populate the data structure underlying the Lowside platform?
Subinoy, would you like to take that?
Yeah, so at this point, we are not assembling any the raw data, but according to the needs of the client, we can see that what is client's necessity, and we can adopt in the Lowside and work collaboratively on that. Because so far we are working on the microbial genomics data sets and all this. So if any transcript of data analysis is there, we can definitely collaborate.
Great. All right, well, we're almost out of time, and actually we're a few minutes over. I do notice that there is a question about the genome standards and what standards we're actually talking about from the Phytobiomes Alliance. And we are aware that there are standards out there. We are more really focused on what regulators need and what the commercial industry actually needs to develop microbial products, for example. So just to mention that. Well, I want to thank Rajesh, Nassir, and Subinoy, excuse me, for that, for your participation today and for a great webinar, and to thank all of our participants. And we will be answering, if any other questions that come in, they will be answered individually back to you. So thank you again, and we hope we see you all on our next webinar. Have a great day.
Thanks to all of you. Thanks, Kelly. Thanks, Isabel.
Thank you, Kelly.
Yeah. Bye-bye.
Bye.
Bye-bye.
Thank you.